Welcome to Virology Conversations, where we break down complex topics into digestible discussions. We take carefully curated scientific sources on a virology topic and generate AI podcast-style conversations. Think of this as your companion for better understanding the most complex topics. Today, we're exploring antibody-dependent enhancement. It's one of those phenomena that seems counterintuitive at first. Antibodies that actually help a virus infect cells rather than neutralize it. This conversation walks through the mechanisms and why it matters. Enjoy!
Welcome to the deep dive. Today we're tackling something really fascinating, but also quite challenging in immunology. Antibody-Dependent Enhancement, or ADE. You've probably heard this term, maybe especially in recent years. It sounds, well, pretty scary. The idea that your own immune system trying to protect you could actually make an infection worse. Yeah, potentially much worse. So our mission today is pretty straightforward. We want to cut through the complexity, really get to the core mechanisms behind ADE. We'll look at the key players inside your cells like FOC receptors and the complement system. And we'll see how this plays out with real diseases. Dengue is a big one, but also coronaviruses. And crucially, what this all means for designing safe vaccines.
Absolutely. And it's important to say up front, ADE isn't some brand new phenomenon tied to, recent viruses. The basic idea that antibodies might enhance infection instead of stopping it was actually first described, believe it or not, back in 1964.
Wow, 1964.
Yeah, by Hawks and colleagues. They were looking at arboviruses. But it really became central to understanding human disease with dengue virus. That's where researchers saw clearly how having some immunity could paradoxically become a major risk factor for severe disease later on. And the key driver we found are antibodies that, while they either don't neutralize the virus effectively, they bind but don't stop it, or they're present at really low levels, what we call subneutralizing concentrations.
So they stick, but they don't stop.
Exactly. That combination binding without neutralizing, that's where the trouble starts.
It really is a biological double cross, isn't it? You rely on these antibodies, and then sometimes they turn cut. So let's unpack that. How exactly does a non-neutralizing antibody make things worse?
Well, it mainly boils down to two different pathways. Think of it like this. The antibody binds the virus, forming what's called an immune complex. And this complex can cause problems in a couple of ways. It might deposit in tissues, causing sort of chronic inflammation, and this is the first major mechanism, it can actually trick certain immune cells into taking up the virus, basically giving it a free ride inside.
Right, enhancing its ability to replicate.
Precisely.
And the sources often split this into two types based on what's happening: extrinsic and intrinsic ADE.
That's right. It's a helpful distinction. Extrinsic ADE is what I just described. The antibody helps the virus get into the cell, enhancing replication directly. We often call this antibody-mediated replication.
Okay, extrinsic is about getting in.
And then there's intrinsic ADE. This is more about enhanced immune activation. Here, the immune complex itself triggers a massive, harmful inflammatory response. It's less about boosting virus numbers and more about causing immunopathology tissue damage.
Got it. Let's stick with extrinsic ADE first, the replication part. You mentioned it tricks cells into taking up the virus. There's a specific receptor involved here, right? The Fc gamma receptor, FCR.
Exactly. The Fc gamma receptor is absolutely key for extrinsic ADE. Think of it as the doorway.
And where do we find these doors?
Primarily on immune cells, macrophages, monocytes, dendritic cells, the cells whose job it normally is to engulf and clear out pathogens or debris.
So a system designed for cleanup gets exploited. How does the antibody act like a key?
Well, the antibody molecule has two ends. One end binds the virus. The other end, the tail or Fc portion, is like a flag. The Fc gamma receptor on the macrophage surface recognizes that Fc tail.
It sees the flag, not what's attached to it.
Precisely. Normally that flag means, hey, I've captured something dangerous, help me dispose of it. The receptor binds the Fc tail and pulls the whole package inside the cell for destruction.
But if the antibody hasn't actually neutralized the virus?
Then the macrophage has just actively imported a live infectious virus. For viruses like dengue or even HIV, which can replicate really well inside macrophages, this is like giving them a VIP pass to their preferred breeding ground. The antibody becomes a Trojan horse.
Okay, that makes sense for viruses that like replicating in those immune cells. But you mentioned intrinsic ADE is different, especially for respiratory viruses. You said it's more about inflammation.
That's right. When we look at things like respiratory syncytial virus, RSV, or potentially coronaviruses, the main concern isn't necessarily that the virus is replicating wildly inside macrophages because of antibodies. that Fc gamma receptor pathway, extrinsic ADE, doesn't seem to be the major issue there.
So what is the issue with intrinsic ADE?
It's the downstream chaos caused by those immune complexes floating around. They trigger another critical part of the immune system, the complement system, and they can send it into overdrive.
Ah, complement. Okay, so the virus-antibody complexes basically set off immune alarm bells.
Exactly. The classical complement pathway gets activated. It starts when a molecule called C1q binds to those immune complexes.
C1q, the first domino.
Right. And once C1q is activated, it sets off a cascade like dominoes falling. This leads to the production of other complement fragments, particularly C3a and C5a.
And those are the problem.
Those are the major culprits in this type of pathology. C3a and C5a are known as anaphylatoxins. Think of them as potent distress signals. They cause massive inflammation, they damage tissues directly, especially delicate lung tissue, and they recruit waves of inflammatory cells, like neutrophils and eosinophils, to the site.
So it's the immune system's response,
Yep.
triggered by the complexes, that causes the severe damage, like acute respiratory distress syndrome, ARDS.
Precisely, or what's sometimes specifically called enhanced respiratory disease, ERD, especially when it follows vaccination. It's this overwhelming complement-driven inflammation that leads to the severe lung injury.
And is there clinical evidence linking complement to severity?
Yes, definitely. For instance, some studies suggest that people deficient in C1q might actually have worse outcomes in certain viral infections, hinting at its complex role. But more directly, high levels of C3a and C5a have been strongly correlated with how sick patients get and even mortality in severe COVID-19.
So the evidence points towards complement activation driving the really bad cases in some respiratory illnesses.
It's a very strong hypothesis, yes. The immune system essentially overreacts, causing more damage than the virus itself might have done.
Let's make this concrete. Dengue is often called the textbook example of ADE, right?
It really is. Dengue showcases extrinsic ADE beautifully, unfortunately. You have 4 main types, or serotypes, DENV 1 to 4. If you get infected with, say, DENV1, you develop strong, lasting immunity, but only to DENV1.
Okay.
You also develop antibodies that can recognize the other serotypes, like DENV2, but they don't neutralize them effectively. They're cross-reactive, but non-neutralizing.
And then if you get infected again, but this time with DEND2.
That's the danger zone. That second, different serotype infection is the biggest risk factor for the severe forms. Dengue hemorrhagic fever, DHF, and dengue shock syndrome, DSS. Those pre-existing non-neutralizing antibodies latch onto the DNV2 virus.
And use the FCC gamma receptors to shuttle it into macrophages.
Exactly, boosting replication. But it gets worse. Inside those infected macrophages, the presence of these immune complexes actually messes with the cell's own defenses. It suppresses the production of type 1 interferon, that's like the cell's main antiviral alarm signal. And at the same time, it ramps up production of anti-inflammatory signals like IL-10.
So it shuts down the defense and promotes tolerance.
In a way, yes. It creates an environment inside the cell that's much more favorable for the virus to replicate unchecked. It's a multi-pronged attack facilitated by the antibody.
Okay, but there was this really strange finding mentioned in the sources from Cuba. It said the longer the time between the first dengue infection, DENV1, and the second one, DENV2, the worse the severe disease was. That seems backwards. Usually, immunity fades over time.
It does seem backwards, but it fits the ADE model perfectly. What likely happened over those years, they looked at intervals from 4 years up to 20 years, is that the neutralizing antibodies, the ones truly protective against DENV1, waned significantly.
Okay, the good ones faded.
Right. But the cross-reactive, non-neutralizing antibodies, the ones that cause enhancement with DENV2, they persisted much longer, or perhaps at levels that became proportionally more significant. So after 20 years, you had this dangerous situation. Not enough neutralizing immunity left to stop DNV2, but plenty of enhancing antibodies ready to make the infection far worse.
Ah, so it's about the balance between neutralizing and enhancing antibodies. You need the neutralizing ones to dominate.
Precisely. High enhancing, low neutralizing. That's the sweet spot for ADE risk.
Okay, that clarifies the dengue situation.
Yeah.
Now, shifting to coronaviruses. Obviously, ADE was a huge concern during the COVID-19 pandemic.
Right.
Was there evidence for it?
Well, the risk was definitely identified early on based on studies with the earlier coronaviruses, SARS-CoV-1 and MERS-CoV. In labs and looking at the virus structures, researchers saw the potential. Pre-existing antibodies, maybe from common cold coronaviruses, could theoretically bind to the spike protein of SARS-CoV-2 but not neutralize it properly.
Leading to potential enhancement.
Right. But, and this is a critical difference from dengue, SARS-CoV-2 doesn't seem to replicate well in macrophages using that FAC gamma receptor pathway. Its main targets are respiratory cells. So that classic extrinsic ADE mechanism isn't thought to be a major driver in COVID-19.
So if ADE is playing a role in severe COVID-19, it's more likely the other pathway.
Exactly. The proposed mechanism, fitting with the clinical data on complement activation, is primarily intrinsic ADE. The idea is that immune complexes form, they activate the complement system excessively.
Leading to that massive inflammation and lung damage we discussed earlier, C3A, C5A, ARDS.
That's the leading hypothesis for how ADE might contribute to severe COVID-19 pathology, yes. It's about triggering that damaging inflammatory cascade, not necessarily boosting viral load in immune cells.
This all leads to a really critical question for public health. Vaccines. If antibodies, which vaccines are designed to generate, can sometimes make things worse, how do we design vaccines safely?
That is perhaps the central challenge ADE poses for vaccine development. Minimizing the risk of ADE is paramount, especially for viruses where prior immunity or cross-reactive antibodies are common. And sadly, we have some harsh historical lessons here.
You mean the RSV vaccine trial?
Yes, the RSV trial from the late 1960s is the tragic classic example. They used a formalin-inactivated RSV vaccine, FI-RSV.
What happened?
When the vaccinated children were later naturally infected with RSV, a horrifyingly high percentage, something like 80%, developed severe respiratory disease, much worse than unvaccinated children. They required hospitalization. Tragically, 2 children died.
That's terrible. The vaccine actually caused enhanced respiratory disease. Why? What went wrong with that vaccine?
The way the virus was inactivated, the formalin treatment, it seems to have altered the viral proteins in a way that skewed the immune response. Instead of generating protective immunity, primarily a Th1 type response.
Th1, that's the good one for viruses.
Generally, yes. Instead, it generated a non-protective, pathological Th2 dominant response. This Th2 response, when the children encountered the real virus, led to an influx of the wrong kind of immune cells, eosinophils, into the lungs.
Eosinophil?
Yes, causing eosinophilic infiltration. Basically, these cells clogged up the lungs, causing severe inflammation and the enhanced disease. It wasn't ADE in the classic antibody entry sense, but rather vaccine-induced immunopathology, a terrible lesson learned about ensuring vaccines elicit the right type of protective response.
So modern vaccine design has to be incredibly careful, not just to generate antibodies, but to generate the right kind of antibodies and the right T-cell backup, avoiding that Th2 skew or antibodies that primarily enhance.
Absolutely. Whether it's dengue, RSV, coronaviruses, or any virus where ADE is a theoretical risk, vaccine developers need to confirm strong neutralizing antibody responses, ideally Th1-biased immunity, and rigorously test to ensure they aren't accidentally creating antibodies that could facilitate Fc gamma receptor uptake or trigger dangerous complement cascades.
We've certainly covered a lot of ground. So to recap for everyone listening, ADE is this double-edged sword. Antibodies can protect, but sometimes they enhance. It happens mainly through 2 routes.
Right. Either enhancing viral entry and replication via Fc gamma receptors, like we see clearly in dengue, that's extrinsic ADE, or triggering excessive damaging inflammation via the complement system, which is the suspected mechanism in severe respiratory diseases like potentially COVID-19, or that RSV vaccine failure that's intrinsic ADE.
And the key players to remember are that Fc gamma receptor, the cellular doorway for extrinsic ADE.
And the complement system, especially C1q starting the cascade and C3A and C5A causing the inflammatory damage in intrinsic ADE. The concentration and type of antibody are absolutely critical.
It really highlights this incredibly fine balance the immune system has to strike between defense and self-damage. Which leads us to a final thought for you to consider. We know some viruses, like dengue, seem to evolve in ways that might even make them better at exploiting these ADE pathways, maybe changes in proteins like NS1. So the question is, in a world now with massive pre-existing immunity from both infections and vaccines against things like respiratory viruses, what evolutionary pressures are we putting on them?
Are these viruses subtly adapting to be better enhanced by the very antibodies we're generating? Are they evolving to navigate our immunity in ways we haven't fully grasped yet?
Something to think about. Thanks for joining us on The Deep Dive.